It's actually a real pleasure to be here for many reasons, and one is that every time the word ecologist has been mentioned in the room, especially when it's been in a non-pejorative way, I've wanted to wave my hand and say thank you for having ecologists in the audience. It's really wonderful for us to have this sort of cross-pollination or interaction together. So the talk that I'll give today is titled Progress Toward Capturing the Biodiversity of Fungal Endophytes, and I am but one piece of a large effort, and I'll be describing our progress over the last couple of years with the list of authors here, including those underlined, actually being a major part of preparing this talk and also being in the room. The group spans multiple universities, and the group that I'm speaking of is defined as the Dimensions of Endophyte Biodiversity Group. This is an NSF-funded project to explore endophyte biodiversity and encompassing with endophytes endolichenic fungi, that is, the fungi that are living in close association with photobionts inside otherwise healthy lichen thalli. So I study endophytes, and over the last 10 or 15 years or so, I've had the good fortune to work primarily in the Western Hemisphere in a more or less latitudinal gradient along the western and eastern portions of North America. So we can see stars here highlighting some of those sample sites where I've gone and collected endophytes from diverse components of the plant community. Through the Dimensions of Biodiversity, or DOB program, we've had the opportunity to expand to a somewhat more global perspective. So our endophyte collections now have spanned the circumboreal belt with the stars that are shown in pink, and that fieldwork is now complete. And that work has generated together more than 51,000 cultures, all of which are maintained as a living voucher collection at the University of Arizona, specifically in my lab. We have, in addition, more than 3,000 host samples for next-generation sequencing, and we're just getting our Illumina data sets back right now. In each of these places, we've had spatially replicated sampling of at least 20 very common species of lichens and plants, and we've done our best to span the green tree of life, encompassing non-vascular plants all the way through angiosperms and many different lineages of lichen-forming fungi as well. I want to just mention the scope of this project extends far beyond sort of the initial discovery phase, and that is to include work in fungal genomics, transcriptomics, molecular phylogenetics, and so on, and especially relevant for today's discussion, biodiversity informatics, and we'll be seeing some of that outcome in the subsequent two talks. Okay. So I was thinking a little bit about how to summarize progress toward discovering the biodiversity of endophytes, and I realized that there are really several components to that. One is the fact that our group is doing this, but another is that, of course, this work is being done around the world by many research groups, and I think the time is uncommonly ripe for us to come together in a new collaborative framework, in particular because to understand the scope of endophyte diversity, as I hope you'll see in part through this talk, we need to have geographic and biogeographic breadth in our sampling, seasonal breadth, host breadth. We need a breadth of different substrates so that we can diagnose the distinctiveness of endophytes relative to other functional groups. We need a diversity of methods to come together. We need shared computational and collections resources, and we need to integrate many different data types together. So it's really easy, obviously. It's something that won't be too hard to do. But the best way to do that, of course, is through collaboration. And in thinking in the simplest way about the question of geographic and biogeographic breadth, I just want to highlight, I made a short database of the geographic origin of the papers on endophytes that I've been asked to review in the last 3 years. And you can see them marked with the stars here. So obviously there's an incredible potential for there to be interactions among these groups, especially because so many of our data are molecular data that will connect to cultured specimens. So there's a chance then to really expand our understanding. So I mentioned that the collection that we have at the University of Arizona, indicated by the little A there at the corner, currently spans about 51,000 cultures, and that these are all living cultures. So obviously I'm here with a plea. This is a fantastic opportunity for a collaboration between an ecologist and systematists who are interested in working with these fungi. So it won't surprise you that the vast majority of these tend to remain as sterile mycelia in culture, so we run into some challenges. However, that hasn't precluded our ability to start integrating these into the fungal tree of life and to gain some new perspectives from that work. Okay? So in using that as a backdrop, I'd like to tell you a little bit about what we found. And often when people think about endophyte studies, I think a lot of the focus is on discovering new tips of the fungal tree, new species. But in fact, what I'll show you is that actually the discovery of endophytes, even using a culture-based approach, and maybe especially using a culture-based approach, gives us the tools that we need to reshape the tree as a whole. So I'm going to use a tree that Jolanta Miadlikowska kindly provided for me. Here we have the backbone for the Ascomycota, and you can see the Pizizomycotina and the Leoshiomyceta. The numbers running vertically then are the number of known described species. So it's generally thought, although there's an interesting debate, and we heard a really nice perspective from Mary Burby today, that the Pizizomycotina may have arisen through the sort of context of interactions between microalgae and photosynthetic prokaryotes and fungi. And then the Leoshiomyceta may, although again with a question based on Mary's talk, may have experienced concurrent diversification with vascular plants. So with that information in mind, it's actually perhaps not too surprising that if we look at the lineages that contain endophytes, those are shown with the boxes here, we see them spread across the Pizizomycotina, and also including here the Pizizomycetes. The black bars are indicating cases in which we don't see known endophytic fungi. And just for fun, because I like to do statistics, I went through and quickly looked at the non-lichen forming lineages and asked the question, do we see greater diversity in those classes that contain endophytes? Now, obviously, the classes that contain endophytes contain fungi that do a lot of other things too. But needless to say, it's actually very exciting from a biodiversity perspective to focus on the lineages that contain endophytic fungi. So in thinking about these from an ecological perspective, there are many different questions that we frequently ask. We're interested in, for example, the degree to which isolation medium or cultural ability will influence our insights about endophytism as a whole. We're interested in biogeographic origins and distributions, and we're also interested in evolutionary shifts. So I want to give you a little bit of a perspective based on our work that I hope will complement the work that's being done by others in the room and beyond on endophytes. So the first thing that I'll talk a little bit about is biogeography. And I thought I would just start with abundance information. And so what I've done here is to show you the data indicating the percent of cultures represented by each class in our collection of endophytes from 5 sites around the circumboreal belt. And so, for example, you can see that we have quite a few Sorteriomycetes and quite a few Pizizomycetes. We can contrast that then with 5 sites across North America, here shown in orange. And then finally, 5 sites across our tropical sampling. And just to give you a sense then of what these abundance curves might actually look like, hopefully you'll be able to see this in the back. There are some little graphs now appearing on the right side of the slide, your right, and the y-axis is the percent representation, the x-axis is latitude. So we're seeing that groups such as the Pizizomycetes and Leoshiomycetes are increasing in abundance as we go farther north. Okay? And the other groups are decreasing in abundance in our samples as we go farther north, that is, they're peaking in the tropics. Okay? So the next thing that we can think a little bit about is what about culturability or the isolation medium that we're using? So in all of our work, we've standardized on a single medium, in part because we had some preliminary data that showed that that medium generated a lot of fungi and a lot of diversity in culture. What's interesting, though, is that we can compare the cultures that we have against next-generation data sets, often taken from the same plant material, and ask, when we do that, do we see changes in the relative abundance of these particular classes? So in the next decoration that will come up on this tree, an orange triangle pointing up will indicate that when we do next-generation sequencing, we tend to get more representation of a class than we do with culturing. And so that has been the case with the Pizizomycetes and the Leoshiomycetes. However, the opposite has been true in our observations with the Sorteriomycetes, the Dithidiomycetes, and the Erochiomycetes. And we just don't have enough data with regard to the other 2 endophyte-containing classes at this time. So it's interesting to see this difference, and it raises some questions about biases that may be implicit both in the culturing and in the next-generation approach. Okay. Finally, I just wanted to say a few words about evolutionary shifts. So several years ago, in conjunction with Yola and Francois, we reconstructed the evolutionary history of many endophytes that we integrated into the phylogeny of the Pizizomycotina, and as part of that, we reconstructed ancestral states and looked at evidence for major ecological shifts in trophic modes. And I'm just going to summarize some of our findings here. Basically, in brown boxes, I'm going to show you classes in which we most often saw interactions or evolutionary exchanges between saprotrophy and endophytes. And in pink, we'll have exchanges between endophytes and pathogens. And so what we can see is that the pizizomycetes and leiotiomycetes in our study were most often connected with a saprotrophic endophyte transition. And that's in contrast then to the dophidiomycetes and urotiomycetes, which more often were connected with an endophyte pathogen transition. And what's interesting is that the sordaromycetes represent an equal representation of endophyte pathogen transitions and endophyte saprotroph transitions. So there's a lot going on here ecologically, and it helps us to set up some hypotheses about biodiversity and the distribution, in part because we can start to see some correlations with next generation sequencing and with abundance patterns that raise some fun questions. So in part, I think I would summarize this part of the talk by saying that we're making progress in understanding the diversity of endophytes, but we're also learning a lot of new things about the questions we actually should be asking. So with this, then, I just want to highlight that among the 51,000 cultures that we have at the University of Arizona, we've done a very rough estimate, an ecologist estimate of the number of species that we have present. And our estimate runs roughly from about 3,000 to about 7,000 species as a minimum estimate for the number of species present. Many of you know that for a lot of ecological work, the unfortunate gold standard at this point is 95% ITS sequence similarity to estimate a species, and we all know that that is a flawed calculation in many ways, but if we use it as a rough estimate, in our case that's almost always an underestimate of the true richness, then I want to show you what we have represented in our collection only at the U of A. So in our collections, we think that we've generated roughly 750 pizizomycete species, 250 leiotiomycete species, over 3,000 sordaromycete species, okay? About 650 dophidiomycete species and 250 urotiomycete species. So it's been estimated by Mary Palm and others that about 95% of endophytes should represent novel species. If that's the case, then truly we have what we hope would be a wonderful resource for systematics. Now, on this tree, I just want to highlight one other group, and that is the xylonomycetes here, for which currently 3 species are known. And this group was actually described by Romina Gassis in conjunction with Francois and with Yola and Priscilla Chiveri. And this group is very interesting. It appears to occur primarily in rubber tree plants, mostly in sapwood, but also in leaves. And this group is important because this was discovered on the basis of a culture-based approach in the context of multi-locus sequencing that was done with those cultures. And as you can see, including it here changes our understanding of this part of the Pizizomycotina tree. So this is one illustration of a way that endophytes can help us understand the relationships of fungi. Okay. So that's at the class level. Let's actually now look at the ordinal level. So here is a very interesting order that was recently described through the work of Koshan Chen, again working with Francois' group. And I'll just sort of cut to the chase here. Here we now recognize that instead of having endophytes in only 2 orders of the urotiomycetes, previously the Urotiales and Chaetotheriales, we now recognize that there's an entire additional order that contains them. Once again, we see that this changes our understanding of sister relationships and other organization in the urotiomycetes tree. And what's interesting is that when these strains were first found, they didn't blast using ITS sequences very well to anything that existed in the database. So there was a little bit of a red flag suggesting that these might be distinct, but the evidence wasn't strong until multi-locus sequencing was completed. And what's wonderful about that is that Koshan then just went on to do some beautiful morphology and to really fit this into a systematics perspective, which is very exciting. And what I also get excited about is the fact that when we look at the understanding of ecology in this order, we see that previously this group was known primarily as pathogens of angiosperms, but all of the green bars here are cases where we're not actually seeing that pathogenic outcome. And instead, of course, we have evidence for endophytism, and in one case, growing inside lichens as well. So this is a case in which we've changed our understanding of ordinal relationships in a major class, even though it's a class that's not especially rich in endophytes. And we've changed our understanding of the ecological modes in that group based on this work. So it's not surprising that in an endophyte talk, of course, I'd want to mention the family Xylariaceae. We had a nice talk earlier today by Mark Stadler about this. Incredible family that really is very rich in endophyte species. And here I just want to highlight some work that's being done by Jana Yu Ren, a postdoc working in my lab, at the family and genus level. her work has looked at more or less reconstructing the relationships of the family using known species, and then integrating into that about 100 species of endophytes and endolycanic fungi that are not represented in any culture collections except ours at the moment. In doing so, she's been able to synthesize the available metadata as well. So I just want to show you a tiny snapshot of her analyses to illustrate how endophytes are contributing in this way. So across the top here, we have 3 words: geography, host, and substrate. And these correspond to categories of metadata. And I'm sure that these are hard to see in the back of the room. So I'll just say that here we have geography. Here we have host information. Is it an angiosperm, a gymnosperm, and so on. And here we have types of substrate, from living lichens and living leaves to soil, insect-associated, and so on. Every time you see a black circle, that's a case in which there is that information available. We know those metadata exist for that exact strain. So what you can see is that here we have many of our endophytes appearing as these green marked species here, okay, in the Daldinia clade. And I should note that this is based on a 4 or 5 gene analysis, depending on the portion of this very large tree. So there are a couple of take-home messages that I'd like to send you with on this. First of all, we have many open circles in our metadata. These open circles indicate a lack of information. In some cases, we don't even know the provenance, the host, the substrate, anything about the ecology based on the existing specimens. So it's going to be very important to do our best as ecologists to assist systematists in filling in that information. The next thing that I want to point out is that this little group here, okay, looks simple, but actually it's quite rich. Each of these green taxa, okay, represents a putative species, a 95% ITS group, okay, that contains many isolates. You can see the number of isolates here. Okay? And so a third thing that we can walk away with is perhaps this gives us a reason to evaluate whether or not truly this specimen identified as Daldinia loculata truly is, given the number of species that intervene between it and the CBS specimen here. So it sets up some phylogenetic hypotheses. So all of that is to say that we're exploring the influx of metadata, how to tap metadata efficiently from publications, and then finally how to match those up with specimens. You'll notice some gray circles here. Those are cases where we're trying to use an exact blast match or a 99% blast match at ITS to gather the metadata. So we're saying, all right, for this taxon in particular, we have information for a close relative, but not that exact species. So we're hoping that by publishing this, we'll sort of call attention to the challenges that we face with metadata. Okay, and then finally, let's go to the species level. So very occasionally, and we actually go out for beer and champagne when this happens, one of our endophytes will offer up some really useful, beautiful characters. And so when we actually have reproductive structures, that inspires us then to do the sort of alpha taxonomy and identify these things, ending up with phylogenies that hopefully will also be related to specimens that are deposited in formal cultural collections. But all too often, this is what endophytes look like, white and fluffy, growing on a plate. And so obviously the challenge that we have then is to describe that biodiversity. Now I showed you those abundance curves, the little plots, the cartoons on the right side of our phylogeny. Those actually mirror our general patterns of biodiversity as well. Many of the groups that we see most predominantly in the tropics, sordiariomycetes, for example, are quite different from the groups that we see most predominantly in the temperate zone or boreal forests, like the pizizomycetes. So we can start to pull all of these things together, in part because of that 51,000 specimens at Arizona, we've sequenced ITS for the vast majority of them, and in most cases, ITS plus an adjacent portion of the large subunit. So work by Yola has allowed us then to, for the first time, take advantage of some of the tools being built by Ignacio to integrate ITS sequences into a single phylogenetic context. And Francois is going to go through in some detail how this works, but basically what I want to show you is that we're looking here at a circular tree where each of the colored tips is an endophyte, and we've included about 400 reference taxa on this tree. So the colors then correspond to the provenance, the origin of the species. And you can see as well that we can test hypotheses. For example, in the Leiotiomycetes, do we have a single origin of endophytism? Okay. Or in boreal forest, do we have a single origin? And using the boreal forest example here, you can see that every time you've got a little blue bar sticking out, that's a case in which we have a boreal endophyte. So it's not consistent with a single evolutionary event and subsequent radiation. Instead, we're seeing these things all around the tree. Okay? So we can zoom in on a couple of classes here, Leishomycetes and Pizizomycetes, and see one of the patterns that makes me all the more interested in these international collaborations, and that is that we have repeated patterns of these local radiations occurring. So here, for example, we see a large group from our North Carolina sampling, here from Arizona. So in the interest of time and keeping our group moving right along, I just want to close with one other example. And that is, of course, finding out about species is a major drive in what so many of us do. But of course, species tells us part of the story, and sometimes we need additional work to help us understand the population level processes that may underlie species diversification. And this too is a place where our role as ecologists can be helpful, and particularly at the interface of population genetics. So here I just want to very briefly mention the work by Ryoko Ono. She worked with Ignacio and with Francois and looked at Lophodermium in pines in the southeastern US. And the take-home message of her work is really shown here. We had a group that was effectively the same species based on our criteria. However, through careful analyses at the population level, it was shown very clearly to contain more than one cryptic species. And in fact, these differ dramatically with regard to divergence times and ecological traits. So in closing, a few things that have emerged in my listening to the talks at this meeting are that, of course, systematics, taxonomy, and nomenclature, and I apologize for putting those all in the same circle, that's the naivete of an ecologist, overlap as a set with ecology. And there are many ways in which ecologists appreciate systematists, and we talk about you at our meetings too, often very nicely. We appreciate that you have this daunting knowledge of fungi. You can identify things that for many of us are not intuitive, and we don't have that knowledge bank. You can help us understand what is a meaningful taxonomic unit. You can help us with curation, help us make sure that we're maintaining specimens that we collect. Because of the power of names, we then have hierarchical knowledge, and that's an incredible contribution so that we can move ecology from the OTU level to a broader perspective. And then finally, that connects to an evolutionary perspective. But I feel that this flows in both directions, and I hope that I've convinced you that ecologists can be your partners in terms of new collections. A lot of us are generating sequence data, and we'd love to share those. We think a lot about metadata, and a coherent effort to share those and analyze those would be wonderful. We can help institute changes in the fungal tree. We can change perspectives on known taxa by populating the tree. We can generate some new hypotheses, and we can also give subspecies perspectives. So hopefully that's a happy circular family that we can all be a part of. And in the middle then, I have the 3 C's. That's a little bit trite, but I would say that those are collaborations, collections, and cyber infrastructure computation, which is sort of the segue to the next couple of talks. So I'll just close then by saying that here I'm actually wearing one other hat, and it's ironic that I would be announcing this as I am an ecologist and not a phylogeneticist per se, but I want to ensure that all of you are aware of this project, and it follows on the Open Tree presentation that David Hibbett gave very nicely yesterday. And that is an NSF-funded program, a 3-year program in the US to support conferences and workshops and hackathons on phylogenetics. It's called Future Phy, for the Future of Phylogenetics. And the idea here is to foster novel integrative data analyses and visualization, interdisciplinary syntheses, and cross-cutting approaches to phylogenetics using lots of data layers, including ecology. And I think that's why I'm involved. I think that's the piece. So if you're interested, please go to futurephy.org and register. You can email us at futurephy.org, contact@futurephy.org, and then you can tweet at futurephy. And then here is the leadership team, including some real systematists and an ecologist. Okay. So my conclusion then, what I hope you'll walk away with is that at Arizona we have somewhere between 3,000 and 7,000, we think mostly novel endophyte species, mostly sterile mycelia, all of them giving up genes. We've been a little slow with our fungal genome sequencing because we've had a hard time getting enough RNA, but we're getting better. This is a subset of what people are collecting around the world. In 10 years of collecting, we've come nowhere near the end point in terms of biodiversity. This is a great resource for collaborative studies. And these are fungi that are not represented in other collections because they are undescribed sterile mycelia. We particularly figure that we'll be enhancing our known richness in the Sordaromycetes and Pizizomycetes, but also across the board. And we have some contributions to the most species-rich classes of the Ascomycota. We hope that this will help shape the fungal tree at multiple levels. As far as the collaborations, I would just close with one picture. This comes from work by Jana Uren in my lab, comparing culturing versus only 500 next-generation sequences. All of our work to generate those 51,000 cultures is depicted by the flatter curve here that is in a cartoon format. You can see, of course, that next-generation sequencing will increase what we have out there. So the time is really ripe to get together. I have many people to thank, including my lab, and I will vacate the stage and give it to Francois. Thank you.